Okay, so in this lesson, we're going to talk about cellular respiration, specifically the very last step, which is oxidative phosphorylation. In yesterday's lesson, we talked about the 4 total steps of cellular respiration, and each of these steps obviously have a million different reactions going on that we're not going to get into, and a lot of enzymes are involved that we're not going to get into. Right, so glycolysis, as we discussed yesterday, is the only step that does not need the mitochondria to happen. Glycolysis happens in the cytoplasm of the cell. It happens outside of the mitochondria. But the other 3 steps, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, those other 3 steps require the mitochondria, right? And so when you eat sugar and you breathe oxygen, this is necessary for cellular respiration to happen. Yesterday, we only talked about sugar. We kept talking about how sugar was being broken down. So sugar was broken down in glycolysis, right? And then we made a pyruvate. Sugar was broken down the pyruvate was then broken down in step 2, pyruvate oxidation, and you get something called an acetyl-CoA, right? And then the last one, citric acid cycle, broke down the acetyl-CoA, and we got the FADH2 and the NADH, right? Let me move this a little bit. In summary, all of the steps, though, it kept breaking down the sugar, and there was also NADH in every single step, right? So the sugar kept getting broken down, and electrons then started getting stored in the NAD plus to form NADH. So in every step, we kept making this NADH, which kept saving the electrons coming from the sugar that kept getting broken down. And then in the citric acid cycle, we made FADH2 as well, right? So what's the point of NADH and FADH2? Well, both of these molecules are going to be necessary for the very last part, oxidative phosphorylation. So you notice we only used sugar so far in steps 1, 2, and 3. So now we're going to also use oxygen in this last step, the oxidative phosphorylation. So how exactly will it be used? So the NADH and the FADH2, they're actually going to donate their electrons to proteins that are on the mitochondria. This is why it's important for the last 3 steps, pyruvate oxidation, citric acid cycle, oxidative phosphorylation. This step has to happen inside the mitochondria because in this very last step, the NADH and FADH2, they donate their electrons now over to these proteins. Where are these proteins found? These proteins are found in the inner mitochondrial membrane. Like we said back when we talked about the endosymbiotic hypothesis, the mitochondria has its own membrane, and it has an additional membrane because it got consumed by the overall cell, right? So when the electrons go to these proteins then transport them, and this is called the electron transport chain again because the electrons are being moved around. Electrons get transported until it reaches oxygen. So remember how we said you bring in sugar and oxygen? That oxygen is necessary in order to receive the electrons from these proteins. Where do these proteins get the electrons? They got the electrons from the NADH and the FADH2. So when the oxygen gets all of these electrons, right, it's going to be negative, and now it's going to be able to attach to 2 H pluses and become water, which is a waste product. So electron transport chain, all it did was transport those electrons from the NADH and the FADH2. It transported those electrons over to the oxygen. And when the oxygen got the electrons, it was negative, and then it's able to form H2O, water, which is a waste product. So this is electron transport chain. Now you might be wondering, well, what's the whole point of transferring these electrons? Only to form water, and water is a waste product. We didn't make ATP. So while it was releasing those electrons, it was also the NADH and FADH2 was also releasing H pluses. So the H plus is pumped through proteins also. So when the electrons are donated, the H pluses are also pumped through between the two membranes. So the H pluses, they're being pumped from inside the mitochondria to between the two mitochondrial membranes. You see how the H pluses, they're between two membranes here. The H pluses are between the outer and inner mitochondrial membrane, right? So all of those H pluses are gathering now. They're gathering in a high concentration in between the two membranes. Why do they need to gather in such a high concentration? If you remember from photosynthesis, when there's a high concentration of H plus, it's acidic, but also the H pluses can now diffuse from a high concentration to low concentration. So the H pluses are going to diffuse back into the mitochondria through the ATP synthase, right? And when the H pluses diffuse through the ATP synthase, this is going to allow the ATP synthase to attach phosphate back onto the ADP and make ATP. So this is where the ATP is made. Okay, so just a quick summary. Electrons were donated because of the NADH and the FADH2. When the electrons were donated, right, the H pluses also come off of those two molecules. And when those H pluses come off, they get pumped so that they're stuck in between the two mitochondrial membranes. And when the H pluses are a high concentration in between the two membranes, now they have a high concentration and diffusion happens from high concentration to low concentration. So the H pluses diffuse back into the mitochondria through the ATP synthase. So this name is called chemiosmosis, okay, when these H pluses are moving through the membrane. So it's just a fancy way of saying, chemiosmosis is just a fancy way of saying the H pluses are diffusing through the membrane. And it's because these H pluses diffuse through the membrane, through the ATP synthase, that's why the mitochondria is able to make ATP again. Okay?